I'm not going to get into a ton of specifics regarding the different panels, but I really wanted to give more general information. So looking at definitions as well as what we do know about epigenetics, and then identifying examples of where gene testing may influence practice. So really looking more on the mental health side, but really at a global picture. So we will talk a little bit about some of the findings, but we're not going to get into detail on those. That could be way more than a 15-minute talk in and of itself for each individual one. So to get started, what is pharmacogenomics? I think this is one of the key buzzwords that's come up quite a bit. So pharmacogenomics is the search for genetic variations that lead to inter-individual differences in drug response, and it's looking at the entire genome. That is different from pharmacogenetics. Pharmacogenetics is also looking for those variations that'll lead to a different drug response, but it's only looking at one particular gene. So it seems like not much of a difference, one gene versus the whole genome, but it can make a tremendous difference. Most of the things that we're going to talk about today are going to fall under the pharmacogenetic differences, so looking at one particular gene. And I thought it'd be helpful to take a step back and do a really brief genetic review, because this will be helpful in the context of talking about epigenetics. So just as a reminder, we have our DNA that is spliced, and it is then transcribed into RNA, which can be translated into amino acids and build out our proteins that then can cause signals and essentially allow us to do all of the things that we do successfully every day. So in thinking about that, as we identify certain genes that are of interest for either drug response or toxicity, the original focus was really looking at single nucleotide polymorphisms, or SNPs. So within that genetic code, I'm going to go back one. Within that genetic code of letters, the original thought was we can find one location where instead of a T, maybe there's a C there instead, and that makes the entire difference. So that's really what we had been watching for. It can happen in a number of different places. As you remember the codons from the mRNA, you actually can have a few different three-letter sequences that then code for the same amino acid. So even though we may find some of those differences, it wasn't resulting in the big differences that we had really hoped that we could find. So this has really forced us to take a step back and look at what are often referred to as GWAS, so the genome-wide association studies. So maybe this one little change doesn't make as much of a difference, but when there's one little change here and one little change here and one little change here, then you see a much bigger difference. So we really had to back up because it wasn't nearly as clear-cut as what we had originally hoped for. So what about epigenetics? Excuse me. So this is the study of mechanisms that turn the genes on and off. So originally we had thought that if we find those changes where the sequence is different, that then we would be able to predict who was going to be, who was going to have blue eyes, for example. What we found is even though they can identify the gene where eye color is coded, if you have the exact same, that's a bad example, but if you have the exact same code, the exact same sequence, it wasn't showing up in absolutely every person like we had anticipated that it would be. So we've actually found that there is a way to turn genes both on and off. Now, in looking back, that really shouldn't be a surprise. We have genetic code in every single cell of our body. And when you're developing, as a fetus, we have cell specialization. So different genes are turned on to help create your eyes and your hair and your nails and your teeth and your liver. So that happens naturally. It happens because it's supposed to. What we're finding is that nurture, or the environment that you're exposed to, can also cause some of these genes to turn on and turn off. So I thought this was a helpful way of thinking about it. Epigenetics is changing the phenotype, so what it looks like, without actually changing the genotype, without actually changing that code. So how can this occur? How does this happen? How can we just turn those on and off? So there's a handful of modifications that have been identified. The DNA methylation is by far the most common and the best understood. So we end up with methyl groups. So if you think way back to your organic chemistry days, your carbon with the 3 hydrogens, it gets attached to the DNA sequence. By doing that, it may make the protein that comes on to separate the DNA unable to attach at that location, which means you can no longer get to that gene sequence. So essentially that would turn that gene off. It may, in other cases, you get the methylation there, it may make it more likely that those proteins can come on and pull the DNA apart so that we can transcribe it into mRNA, so it would turn them on. So depending on where they are, it could do either one. The other 3 are not as well defined, but I do think it's important because there are multiple ways that this could occur. So chromatin remodeling. So chromatin is actually the way that our DNA is packaged within the cells themselves. So you have DNA that's packaged with proteins, and it's called a histone. Now, if the histone is of a different size, it may make it more or less likely that it could be accessed by those other proteins to be turned on or turned off. The chromatin remodeling, all of those histones that are linked together, if they're compressed, which they are most of the time when they're in storage, we can't access those genes. It needs to be kind of pulled apart or strung out, kind of like if you're knitting or crocheting, you can't have all of the yarn in a big ball at the end. You actually need to separate it so it's in a long line. So if the chromatin is remodeled in a way where that packaging is really loose, then the proteins can access the DNA material much easier. If it's packed really tight and it's really tightly wound, then it's less likely that the proteins can access that genetic material. So both of those do seem to be involved with epigenetics. And we'll talk about some of the prenatal and immediate postnatal factors that seem to be involved with those. There's another mechanism, so non-coding RNA mechanisms. So parts of the DNA sequence that don't necessarily code for RNA that may essentially terminate the transcription early so that you're not getting the full message. So maybe it results in a protein that's not functional anymore. So they're still trying to understand that one. That one seems to be the one that's, we don't know what all of the genetic material codes for just yet. And that's kind of in that gray space there. But for completeness sake, I wanted to include them all. All right, so we know that nature, now that I swallow my cough drops, so I'm not afraid that it's going to spit out. So nature can cause cells to specialize, and it turns on and turns off genes, but nurture can as well. So what exactly is it that, that is that nurture? So it's a number of different environmental factors. So prenatal conditions, and I found this quite interesting. So a mother's diet, the amount of stress that she's under, if she gets ill at any point during pregnancy, the environmental toxins that she may be exposed to, all seem to play a role in epigenetics. The father's diet may also play a role in epigenetics. So up to a minimum of 6 weeks prior to conception, if the father's diet is relatively high or low in particular items, so they're looking at vitamin B12 and folic acid, it may actually impact how well the baby is able to perform on cognitive exams. So they've done most of this testing in animal models, but it seems to have at least some correlation in humans as well. So it's not just the mom, it would be both the mom and the dad. So that's in the developmental process. There can be early postnatal factors as well. So if there is, if it was a very stressful birth, if there is any type of bonding issue or neglect, initially that can play a role, as well as how well the baby is able to feed right away. All seem to also put the young brain under stress and may trigger some of these signaling mechanisms. The more that they're looking at it, initially it was thought that this happens early in life, and then it has longer term effects. What they're finding is these changes can continue to happen over the lifespan. So we've identified where they can continue to happen and it's detrimental. They're also looking at ways to be able to reverse them, and we'll talk about that in just a minute on the next slide. So the availability of a balanced diet. The two main vitamins that they've looked at are B and D, but a relatively robust diet overall. They're also looking at differences for Atkins type diets. So if you have a relatively high protein, low carbohydrate diet, what might that turn off or turn on? Because that seems to play a role. The environmental pollution that individuals are exposed to, and this could be anything from lead-based paint, to different particulates in the water source, to the air particulates that individuals are exposed to. And the last one is much more generic, but it's stressors. So anything that would cause stress upon the individual human. So stress that then results in inflammation, and that inflammation seems to be what triggers some of those mechanisms to create the epigenetics, to switch genes on or switch genes off. And it's not consistent. That's why you can see differences between twins in the way that they actually respond to the same stressor. So it's not something that's consistently applied. So what can we do on the protective side? So definitely a balanced diet, making sure that all of the vital nutrients, vitamins, and minerals are included from as early on as we can throughout the entire life cycle. Meditation, so there are a handful of studies that are looking at the role of meditation in trying to be protective for some of the epigenetics. So they found that for individuals who routinely participate in meditation, they tend to have lower biomarkers of inflammation. So like their C-reactive protein would be lower, and some of the other interleukins or cytokine signals are also lower, which seems to reduce the incidence of some potentially negative consequences. So obesity, cancer, other stress-related conditions. It also seems to protect the telomeres. So those are kind of the buffers that are on the ends of the genetic material. So if you think of them almost like a cushion, like the cushion that you have, I'm looking at it right now, on the wall to protect the wall from the door slamming into it. So you have these buffers as well, and over time your telomeres get smaller and smaller. Meditation seems to be able to maintain them at a more consistent and robust level so that they're a better cushion for your genetic material. The last activity is, the last protective feature would be physical activity. So again, thinking about reducing inflammation overall, really kind of promoting those healthy telomeres and being beneficial for pretty much every organ in your body. They did not necessarily specify whether this needed to be more cardio type activity versus strength training, but just general physical activity on a consistent basis overall. Although there is a little bit of research to suggest that intensive training, so like individuals who are relatively far on the spectrum of doing intense training, ultra marathons, things like that, may actually start to do some damage because of the inflammation long term. So where could this be beneficial? The goal really is to have individualized medicine. And I think most of the places, especially in the lay media that you look, this is the direction that everybody's being encouraged to go into. We would like to have the genetic materials so that we could have this white group down in the corner, a drug that's not toxic and it's beneficial. So we know exactly what to give you that's going to work and that's not going to cause you side effects. We're not there yet. So we still have drugs that are not toxic and not beneficial. We definitely would want to try to avoid those. And I'll say we're probably farther along with identifying this group than we are with some of the other ones. A drug that's toxic, but it's still beneficial, and a drug that's toxic and not beneficial. So we're still kind of muddling through those other categories. But that's the goal, and that really is, if you look at a lot of the material that's available just on the internet, I would say that's how it's being sold to most of our patients. I've had an instance not long ago where I tend to be more on the skeptical side. I do think we need to be an advocate for our patients to help provide the education, because it's not that simple. Because of the epigenetics, because your genetic code may not be the way that you actually look, your phenotype may not match that. We're not nearly as close to this as I think some of the marketing may lead people to believe. So what do we actually have? So I'm going to start with the stuff that has more robust data. And I would say our pharmacokinetic considerations are actually much farther along. So we have FDA product labeling for the pharmacogenetics of all of these different medications. So they've identified polymorphisms in the genetic code that codes for the cytochrome P450 enzymes, and they've recognized a difference in these particular groups that may require a dosage change. The 2D6 is by far the one that has the most robust data. So this is located on chromosome 22, and it's very, very polymorphic. So there's more than 120 allelic variants. So you can get a tremendous amount of variability. So in these phenotypes that we can see, depending on what sequence they have, a patient can be an ultra-rapid metabolizer. So they have multiple copies of the most efficient 2D6 enzyme. So they can chew through 2D6 substrates incredibly quickly. It doesn't mean we can't use those drugs, but it means we need to raise the dose much higher because they're able to process a lot more. The extensive metabolizers, so they may have a couple copies of a relatively efficient version of the 2D6 enzyme. An intermediate metabolizer, in this case, this is going to be a middle-of-the-road person. So you've got functional copies. So you may have one copy of a really efficient enzyme, or you may have a couple copies of a less efficient enzyme. And then we have our poor metabolizers. And in some cases, this is an enzyme that doesn't work very well, that's relatively inefficient. In other cases, it may be pretty much absent, so they really don't have a functional 2D6 enzyme. So because of that, we can see relatively significant differences. In poor metabolizers, we see that in about 5 to 10% of Caucasian patients, and less than 1% of Asian patients. So for whatever reason, it's more likely to occur in patients of Caucasian descent. Our intermediate metabolizers is the predominant phenotype of patients of Asian descent, and it's one substitution of an amino acid that can impact the amount of activity, how efficient that enzyme is. So that may be one base pair different that makes that difference. So in this case, we're really looking at the fine details. And then our ultra-rapid metabolizers, so this has been found in about 10 to 15% of Caucasian patients. So when you see some of the genetic, a large number of the genetic testing results, they'll have those red, yellow, and green columns. And it tends to correlate with the amount of activity that they have. So just because somebody's a poor metabolizer of a 2D6 substrate, risperidone may show up in the red category. That does not mean we cannot use that. But I think that's really difficult from an educational standpoint, because the visual is, hey, stop, this is no good, red is danger. So it's really important that we're aware of that. A poor metabolizer means that you're not able to process it, so we should start low, and we'll need to maintain at a relatively lower dose and be mindful of watching for those side effects. So it's not a mutually exclusive. So the cytochrome P4 enzymes definitely is one area that we have some good, robust data to make some patient decisions on. The other area is in predicting toxicity. So I'm going to call out specifically the risk of Stevens-Johnson syndrome. So this particular potentially fatal condition has been identified to be linked with the human leukocyte antigen and a couple different sites on this particular gene. So they're both listed here. So HLA-B, the 1502, and then the 5801. So for patients who are of Asian descent, before they start a carbamazepine or carbamazepine-related product, it is recommended that we test for this particular polymorphism. So this test result should be received before initiating treatment. And that's because if they have this polymorphism, they're much more likely to have this potential adverse effect, which could be incredibly problematic and potentially deadly. They've also found it a little bit with allopurinol, but they don't have as clean of a demographic. It doesn't seem to be quite as predictive in Asian patients, but they do recommend some testing for the allopurinol as well. So in predicting some of the testing, this could be beneficial. How about looking on the other side in predicting response? This by far does not have consistent, robust data. So we have a large amount of reports that polymorphisms in receptor subtypes may be predictive of response. So some of the serotonin 2C receptors that may show whether or not a patient may be more likely to gain weight with a second-generation antipsychotic. In some cases, it seems to be associated with an increased response to an anti-anxiety medication, so like an SSRI. But it is not consistently applied. So we don't have enough of a robust data set to say, yes, this is the test to say that you'll respond to citalopram for generalized anxiety. For dopamine, there's a little bit better data with some of the dopamine receptors. There seems to be a polymorphism that's associated with kids that don't respond to stimulants. Again, it's not consistently seen. We don't have a huge database to draw off of, but there may be some benefit there that you'd be able to identify those kids that aren't going to respond to a stimulant. The argument is it cost effective? At this point, no, it hasn't been seen to be cost effective just yet. Why? Because most kids can tolerate stimulants, and the amount that you're talking about that don't respond to stimulants are less than 5%. So trying to identify just that group, it's not necessarily detrimental to have uncontrolled ADHD, and you would get there relatively quickly because we see a relatively immediate response from those stimulant medications. There's also some interesting research in looking at other types of neurotransmitters. Acetylcholine I'm going to call out here because they've done quite a bit of work looking at some acetylcholine receptors in patients who may be at higher risk for developing tardive dyskinesia. So especially in kids who may be sensitive to this, this might be a marker as we follow that you'd want to have that information to know whether a kid would be very sensitive to an antipsychotic medication, and then maybe we would steer away from those that are more likely to cause tardive dyskinesia. So something like risperidone, maybe opt for something more like aripiprazole. There are other genetic locations that we don't have as clear an idea of exactly what they do. So we have a location, and they may be involved with secondary messengers, they may be involved with other enzyme affinities, they may be involved with thresholds, so how sensitive a neuron may be or not be. In cases like panic disorder or seizures or even bipolar disorder, how sensitive those neurons are and how quickly they would fire, if we can impact that, that might make a big difference. But it's relatively inconsistent. So we don't have enough information to be able to say that, yes, we have these locations that will definitively tell you what you should do as far as treatment's concerned. They are looking very, very intensely at a number of different medications. And what you'll see is all of these genetic locations and then all of the antidepressants or all of the antipsychotics, because they're trying to predict the ability of this polymorphism at this location, and is that going to respond? There are some that look like they have relatively robust effect sizes, but then you look at another collection of studies and they did not find any difference whatsoever. Again, that's most likely due to the epigenetics. So it may not just be that one location, there may be 5 that are involved. And when everything aligns just right, then you can predict response, but we just don't have that amount of data or support at this point. So my take home is genomic testing and genetic testing can definitely give you additional information. And in some cases, that may be something that you're hoping for. Guiding treatment decisions, pharmacokinetically or safety-wise, especially with Stevens-Johnson syndrome, absolutely. We have enough data to be able to support that at this point. To predict an efficacy response, we are not there yet for any of the mental health conditions. There are suggestions, and we're definitely moving in that direction, but we don't have that at this time. And that's what I've got.